Similia similibus curantur, is the most fundamental concept of homeopathy. It is the basic theoretical foundation upon which the whole superstructure of this therapeutic system is built up. Even though homeopaths consider it as a natural law of therapeutics, critics of homeopathy never accept such a law or pattern really exists in nature. They use to portray it as a natural fallacy of Hahnemann. When attempting to establish homeopathy as a scientific medical system, it is essential that we should be capable of providing a scientifically plausible explanation for the biological mechanism of cure involved in similia similibus curantur, and prove it according scientific method. Samuel Hahnemann, great founder of homeopathy, says that a substance can cure a disease, if the symptoms produced by that substance in healthy individuals are similar to the symptoms expressed by the person in disease condition. Looking from a scientific perspective, similarity of symptoms indicates similarity of affected biomolecular pathways, similarity of molecular inhibitions, similarity of target molecules, similarity of involved drug molecules and pathogenic molecules, and ultimately, similarity of their functional groups. In order to be capable of explaining similia similibus curantur, scientifically, first of all, we have to study carefully the phenomenon known as competitive inhibitions in modern biochemistry. As all of us know, competitive inhibition is the interruption of a biochemical pathway owing to one chemical substance inhibiting the effect of another by competing with it for binding or bonding with same targets, due to the similarity of their functional groups. Several classes of competitive inhibition are especially important in biochemistry and medicine, such as the competitive form of enzyme inhibition, the competitive form of receptor antagonism, the competitive form of antimetabolite activity, the competitive form of poisoning, etc. In competitive inhibition of enzyme catalysis, binding of an inhibitor prevents binding of the natural target molecule of the enzyme, also known as the substrate. This is accomplished by blocking the binding site of the enzyme, also known as the active site, where the natural ligands or substrates are expected to bind with. Competitive inhibition can be overcome by adding more substrate or natural ligands to the reaction, which increases the chances of the enzyme and substrate binding. This is known as reversibility of competitive inhibitions. Most competitive inhibitors function by binding reversibly to the active site of the enzyme. As a result, many sources state that this is the defining feature of competitive inhibitors. In competitive inhibition, an inhibitor having functional groups similar to the normal substrate or ligand binds to the enzyme, usually at the active site, and prevents the substrate from binding. At any given moment, the enzyme may be bound to the inhibitor, the substrate, or neither, but it cannot bind both at the same time. During competitive inhibition, the inhibitor and substrate compete for the same active site. The active site is a region on an enzyme which a particular protein or substrate can bind to. The active site will only allow one of the two complexes to bind to the site, therefore either allowing for a reaction to occur or yielding it. In a state of competitive inhibition, the inhibitor molecules resemble the substrate and therefore take its place, thereby binding to the active site of an enzyme. Increasing the substrate concentration would diminish the competition, and help the natural substrate to properly bind to the active site and allow a reaction to occur. When the substrate is of higher concentration than that of the competitive inhibitor, it is more likely that the substrate will come into contact with the enzyme's active site than the inhibitor. Methotrexate is a chemotherapy drug that acts as a competitive inhibitor. It is structurally similar to the coenzyme called folate, which binds to the enzyme dihydrofolate reductase. This enzyme is part of the synthesis of DNA and RNA, and when methotrexate binds the enzyme, it renders it inactive, so that it cannot synthesize DNA and RNA. Thus, the cancer cells are unable to grow and divide. Another example of competitive inhibition involves prostaglandins, which are made in large amounts as a response to pain, and can cause inflammatory process. Essential fatty acids form the prostaglandins, and when this was discovered, it turned out that these essential fatty acids are actually very good inhibitors to prostaglandins. These fatty acids inhibitors have been used as drugs to relieve pain because they can mimic as the substrate, and bind to the enzyme, and block prostaglandins due to their similar functional groups. An example of non-drug-related competitive inhibition is in the prevention of browning of fruits and vegetables. For example, tyrosinase, an enzyme within mushrooms, normally binds to the substrate, monophenols, and forms brown O-quinones. Competitive substrates, such as certain substituted benzaldehydes for mushrooms, compete with the substrate, lowering the amount of the monophenols that bind. These inhibitory compounds added to the produce keep it fresh for longer periods of time by decreasing the binding of the monophenols that cause browning. This allows for an increase in produce quality as well as shelf life of mushrooms. Competitive form of enzyme inhibition, the competitive form of receptor antagonism, the competitive form of antimetabolite activity, and the competitive form of poisoning. Ethanol C2 hydrogen hydroxide serves as a competitive inhibitor to methanol and ethylene glycol for the enzyme alcohol dehydrogenase in the liver when present in large amounts. For this reason, ethanol is sometimes used as a means to treat or prevent toxicity following accidental ingestion of these chemicals. Strychnine acts as an allosteric inhibitor of the glycine receptor in the mammalian spinal cord and brain stem. Glycine is a major postsynaptic inhibitory neurotransmitter with a specific receptor site. Strychnine binds to an alternate site that reduces the affinity of the glycine receptor for glycine, resulting in convulsions due to lessened inhibition by the glycine. After an accidental ingestion of a contaminated opioid drug desmethylpridine, the neurotoxic effect of 1-methyl-4-phenyl 1,2,3,6-tetrahydropyridine MPTP was discovered. MPTP is able to cross the blood-brain barrier and enter acidic lysosomes. MPTP is biologically activated by MAO-B, an isozyme of monoamine oxidase MAO which is mainly concentrated in neurological disorders and diseases. Later, it was discovered that MPTP causes symptoms similar to that of Parkinson's disease. Cells in the central nervous system astrocytes include MAO-B that oxidizes MPTP to 1-methyl-4-phenylpridinium MPP, which is toxic. MPP eventually travels to the extracellular fluid by a dopamine transporter, which ultimately causes the Parkinson's symptoms. However, competitive inhibition of the MAO-B enzyme or the dopamine transporter protects against the oxidation of MPTP to MPP. A few compounds have been tested for their ability to inhibit oxidation of MPTP to MPP, including methylene blue 5-nitroindazole, noramine, 9-methylnoharmine, and monadione. These demonstrated a reduction of neurotoxicity produced by MPTP. Sulfanilamide competitively binds to the enzyme in the dihydropteroate synthase DHPS active site by mimicking the substrate para-aminobenzoic acid PABA. This prevents the substrate itself from binding, which halts the production of folic acid, an essential nutrient. Bacteria must synthesize folic acid because they do not have a transporter for it. Without folic acid, bacteria cannot grow and divide. Therefore, because of sulfur drugs' competitive inhibition, they are excellent antibacterial agents. An example of competitive inhibition was demonstrated experimentally for the enzyme succinate dehydrogenase, which catalyzes the oxidation of succinate to fumarate in the Krebs cycle. Malonate is a competitive inhibitor of succinate dehydrogenase. The binding of succinate dehydrogenase to the substrate, succinate, is competitively inhibited. This happens because malonate's chemistry is similar to succinate. Malonate's ability to inhibit binding of the enzyme and substrate is based on the ratio of malonate to succinate. Malonate binds to the active site of succinate dehydrogenase so that succinate cannot. Thus, it inhibits the reaction. Competitive inhibition can be reversible or irreversible. If it is reversible inhibition, then effects of the inhibitor can be overcome by increasing substrate concentration. If it is irreversible inhibition, the only way to overcome it is to produce more of the target, and typically degrade, or excrete the irreversibly inhibited target. In virtually every case, Competitive inhibitors bind in the same binding site or active site as the substrate, but same site binding is not an essential requirement for competitive inhibitions to happen. A competitive inhibitor could bind to an allosteric site of the free enzyme and prevent substrate binding, as long as it does not bind to the allosteric site when the substrate is bound. For example, strychnine acts as an allosteric inhibitor of the glycine receptor in the mammalian spinal cord and brain stem. Glycine is a major postsynaptic inhibitory neurotransmitter with a specific receptor site. Strychnine binds to an alternate site that reduces the affinity of the glycine receptor for glycine, resulting in convulsions due to lessened inhibition by the glycine. Actually, it is this phenomenon of competitive inhibitions that works behind Similimum concept of homeopathy. It actually means, a molecular inhibition produced by a particular pathogenic molecule could be removed by utilizing a drug molecule having competitive relationship with it due to the similarity of functional groups. If the functional groups of pathogenic molecules and drug molecules are similar, they can bind to similar molecular targets and produce similar symptoms. That is why homeopathy tries to identify similarity between pathogenic molecules and drug molecules by observing the similarity of symptoms they produce. Through the principle of similia similibus curantur, Hahnemann was actually trying to explain and utilize this phenomenon of competitive inhibitions for the purpose of developing his new therapeutic method. When we try to remove pathological molecular inhibitions by using competitive inhibitors as done in allopathy, there is always a chance for developing new drug-induced diseases due to their off-target actions. This phenomenon underlies the dangerous side effects of most of the chemotherapeutic drugs. It means, when we use molecular forms of similimum or competitive inhibitors for treating a disease, it may lead to establishing new diseases that may be more harmful to the organism. Hahnemann also observed this possibility of drug-induced diseases, and he tried to overcome this danger by using potentized forms of competitive inhibitors or similimum. In order to overcome this adverse effects of competitive inhibitors when used for therapeutic purpose, Samuel Hahnemann developed the technology of drug potentization. Homeopathic potentization involves a process of preparing molecular imprints of drug molecules in water-ethyl alcohol medium using drug molecules as templates. Molecular imprints are supramolecular clusters formed in the imprinting medium, wherein the spatial conformations of template molecules remain engraved as nanocavities or intermolecular voids. Due to complementary conformations, these molecular imprints of competitive inhibitors can act as artificial binding pockets for the pathogenic molecules and deactivate them, thereby removing the pathological molecular inhibitions they had produced in biological molecules. If symptoms produced in healthy individuals by a drug substance appear similar to the symptoms expressed in a disease condition, it obviously means that the particular drug substance as well as the particular disease-causing substance contain some chemical molecules having similar functional groups or moieties, so that both of them were capable of binding to same biological targets in the organism, producing similar molecular errors that are expressed through similar trains of symptoms. Molecular imprints of similar chemical molecules can act as artificial binding pockets for similar chemical molecules, and deactivate them due to their mutually complementary conformations. It is obvious that Samuel Hahnemann was observing this phenomenon of competitive relationship between substances in producing similar symptoms by acting upon living organisms. Pathogenic molecules produce diseases by competitively binding with the biological targets by mimicking as the natural ligands due to their conformational similarity. Remember, Hahnemann was working during a period when modern biochemistry has not even evolved. It is obvious why Hahnemann could not explain the phenomenon he observed using the paradigms of modern biochemistry. But his extraordinary genius could foresee its implications in therapeutics. Once we could convince the scientific community that similia similibus curantur is based on the natural phenomenon of competitive relationship between chemical molecules having similar conformations in binding to the biological molecules that is well explained in modern biochemistry, homeopathy will be inevitably recognized as science.